dense, so they are more sensitive to cause mutation. So if we'll see the evolutionary development of two species, and if we'll compare the structure of the DNA of the same species and the RNA, then it is possible that with time DNA might be changed due to the mutation due to its weak structure, while RNA will never be changed due to its very strong resistant structure against any changes. So instead of DNA, RNA is more preferred in the studies. So ribotyping is similar to probing restriction fragments to chromosomal DNA with clone probes. So that probes either selected specifically or randomly cloned probes, or it can be specific such as virulence factor or antigenic factor, etc. Any probe we can consider, and fingerprintings are made. So it is a method that can identify and classify bacteria based upon differences in RNA. So it generates a very highly reproducible and precise fingerprint that can be used to classify bacteria from the genus through and even beyond the species level. So naming is also important. As we have our identity, then microorganisms also need their identity. So various scientists have performed the classification, identification, but to provide some special space, some reference, they should be quoted somewhere, so as in future also we can compare them, we can study them. So for such a purpose there should be some manual, and such type of one and only manual which is prepared in case of bacteria is known as the Bergey's manual. So now we'll see some of the chronological development in the publications of this Bergey's manual. So the Bergey's manual of systematic bacteriology is the main resource for determining the identity of bacterial species, utilizing every characterizing aspect to classify them. This is David Bergey. He is the pioneer of Bergey's manuals. Now the chronological development, then the first Bergey's manual was published in the year 1923. Until 1936, the Bergey, he set up an educational trust. It was the non-profit trust, and that Bergey's manual, it ensures that it will be a self-perpetuating publications. After the first publication of Bergey's manual under the auspices of Society of American Bacteriologists in 1923, it soon became a most widely accepted system which can be used to identify bacterial isolates in the laboratory. So Bergey's manual, from the first edition in 1923 to 8th edition in 1994, it was known as Bergey's manual of Determinative Bacteriology, and from 9th edition the title was changed to Bergey's manual of Systematic Bacteriology. To reflect why the title was changed, it is to reflect an increased coverage of bacterial characterization, classification and taxonomic problems in addition to that identification aspects. So here we can see the basic comparison between the two manuals, the Bergey's manual of Determinative Bacteriology versus Systematic Bacteriology. So the purpose of Determinative Bacteriology was to provide identification scheme for identify to bacteria as well as Archaea, while the Systematic Bacteriology provides not only this identification schemes but also the phylogenetic information of bacteria and Archaea. Then why these two are different? Because the Determinative Bacteriology was based only on morphology or differential staining or biochemical tests only, while Systematic Bacteriology is based on the genotypic study, that is rRNA sequencing method. So if we'll see some of the details, then the Determinative Bacteriology has 5-volume resource and it covering all the prokaryotes. So classification is also based on phylogenetic information and morphology and identification. Two main domains are considered here. One is Archaea and second is Bacteria. The whole determinative bacteriology is further divided into 5 major subgroups with 25 different phyla. The volume 1A, which contains the domain Archaea, means they are the very primitive microorganisms and showing extreme habitats as well as extreme modes of nutrients. The volume 1B, which contains the general, routine, ordinary bacteria which are observed. Volume 2 to 5 are further divided into various phylum, as we can see on the screen. The Phylum Proteobacteria, or the Phylum Firmicutes, or the Phylum Actinobacteria. Now we'll see the details of these domains. So first we'll see with the domain Archaea. It is further divided into 3 basic groups: Crenarchaeaota, Archaeaota, and Archaeaota. Crenarchaeaota means they are the most thermophilic archaea. What is their specificity? Means they can use sulfur compounds as electron donors as well as electron acceptors. It is not compulsory that they are thermophilic always. Archaeaota means it mainly includes the methane utilizing methanogens or salt utilizing or salt loving microorganisms like halophiles or even sometimes thermophiles. And Archaeaota is the group of archaea which are isolated, but still nowadays we are not capable of cultivating them into the artificial laboratory environment. Now the phylum which are included in the volume 2 to 5, it mainly includes various groups like the purple phototrophic bacteria or the nitrifying bacteria which fixes nitrogen, or the sulfur and iron oxidizing bacteria, hydrogen oxidizing bacteria, methanotropes or methylotropes, pseudomonas, acetic acid producing bacteria, or free living and nitrogen fixing bacteria. These are some of the another list like the Neisseria or enteric bacteria, Vibrio, Rickettsia, Spirilla, or even sulfate and sulfur reducing proteobacteria. Now another phylum which is known as the Firmicutes. So it generally contains non-sporulating, means the spores are not formed and having low G plus C percentage ratio. They are the gram positive bacteria. Firmicutes mainly includes gram positive bacteria, or sometimes it might include cell wall lace or with low G plus C or gram positive bacteria like mycoplasmas. Now Actinobacteria, the last phylum, as we can see in the determinative bacteriology, having high G plus C and gram positive bacteria, or filamentous or high G plus C containing gram positive bacteria, or we can say the actinomycetes like Streptomyces or any other actinomycetes group. Now we come to the Bergey's Manual of Systematic Bacteriology. So it is published in the year 1984, though the determinative is still published as a guide for identifying the unknown bacteria. Nowadays Bergey's Manual of Systematic Bacteria is more precisely used. Then how it is formed? So the change in the volume set to systematic bacteriology came in new contract in 1980, whereupon new style included relationships between organisms and expanded scope overall. So this new style was picked up for a 4-volume system, and the publication was done in year 1984. So the Bergey's Manual of Systematic Bacteriology was divided in 4 volumes. The volume 1, which is known as the Gracilicutes, it includes mainly the gram negative bacteria. Volume 2 is known as the Firmicutes, which mainly includes gram positive bacteria. Volume 3, which is generally known as Tenericultes, it includes some of the slightly different gram-negative bacteria along with Archaea, and volume 4, which is known as Mendosicutes, it includes generally the filamentous actinomycetes. So what is the current status? So as the newer identification schemes are applied, or the newer classification systems derived, or the newer information derived on the base of 16S rRNA nucleotide sequencing, some of the newer microorganisms are added. So the current grouping or the volumes of systematic bacteriology is changed from the previous edition. So it now, instead of that 4 volume, it contains 5 different volumes. Volume 1, which was published in year 2001, it contains mainly the information regarding the Archaea and the deeply branching phototrophic bacteria. Volume 2A, it contains mainly the introductory assays. Volume 2B, the the information regarding gamma proteobacteria, and volume 2C, which includes other classes of proteobacteria. Now volume 3, it mainly contains the Firmicutes, means the gram-positive bacteria-like, and volume 4, it includes the spirochetes or Tenericultes or even about Mollicutes. And the last volume remains same, it mainly contains the information regarding the actinobacteria. So this all is about the various systems, about classification, identification, but with it the naming is also important. So to identify some newly discovered organism, we must have some identity. Like humans, we are also known by some names, then microorganisms should be known by some names. So in classification, binomial nomenclature is also very much important part of the classification, because we have to give some identity to that particular microorganisms also. So binomial nomenclature system was first given by the scientist Carl Linnaeus. So some of the basic rules are applied to that nomenclature. Why this is known as the binomial nomenclature? Because it mainly consists of giving two names, as we are having one name and one surname. Same way, microorganisms or even bacteria, if we are talking, then they are divided in two parts. One is the genus name and second is the species name. So together, by combining this, the system is known as binomial nomenclature. Now what are the rules given, how the names are given, or how we can read their name? There are some rules of the binomial nomenclature which we can see on the screen. So the first rule is the genus comes before species. So in case of Escherichia coli, Escherichia is the name of the genus and coli is the name of the species. So genus name is always capitalized, the first letter of the genus should be capitalized, and the species name should never be capitalized like coli. So both the names are always either italicized or underlined. The genus name may be used alone like Escherichia, but species name never, like we cannot write coli. The genus name may be abbreviated, but for the abbreviation it must be used with the species name like E and then the name of the species like coli, means the E. coli. Or sometimes the genus abbreviations are not only used, means we cannot use only E without the conjunction of the species name. Okay. Abbreviated the first letter of the genus, then it should be unambiguous, means the same letter cannot be covered by some other genus. So these are some of the rules of binomial nomenclature. So this is also the part of classification. So this ends our discussion. We have used various references of course, all the volumes of Bergey's Manual of Systematic Bacteriology and the volumes of Determinative Bacteriology, as well as the Black and also the Pelzar by the Michael J. Pelzar, Microbiology. So, at the end, I would like to thank the SANDHAN, an initiative of live lecture series initiated by the Department of Higher Education and Government of Gujarat. Also, we are very thankful for the very supportive nature and help of the BISEC studio members. And then
we are also thankful, obviously, to the principal of our institute, Dr. S.C. Parikh, who has given us this opportunity to come out of our classrooms, institutional classrooms, and face a huge viewership crowd of our students all over the state. We are also thankful to the staff members of the department of our institute and the students who are our best audiences. I'm thankful to all the members who are directly or indirectly cooperating with us to deliver this series of lectures. Thank you.